Probably finish this topic on Tuesday and look at some of the other things. Now, I'll tell you where it ends. I don't know myself yet, but we have this in here. Okay, well, let's take a look at this process. This has been a hot button topic in cell and molecular biology now. Okay, the process known as did not curse. The process known as RNA interference. Should be all the put on YouTube. A piece of chalk breaking and then a Anglo-Saxon phrase or two. And then everybody in the world can see me cursing in class. I don't want to do that.
What kind of chalk is good chalk? Like, what kind do you like?
Quality chalk that doesn't disintegrate when you use it. I don't know what brand this is, but Crayola chalk is terrible. I used to call it crapola chalk because that was a reflection as well. They may be great for kids' grades. For chalk, they're absolutely abysmal.
Well, our organic teacher was talking the other day, and she was like, it's the rose art chalk that she didn't like.
Stuff is called Prang. Probably made in China or something. Sounds like that. Prang immersive. Made in France. Okay, France made a great cuisine, but chalk they really leave that to the English to get hold.
Have you had good chalk?
Yeah, some years back before you started switching to the old cheap crapola chalk. Yeah.
Not many people make chalk though.
Excuse me?
I said not many people would make chalk now since most people have gone to whiteboards and stuff.
Yeah, all those whiteboards are terrible. I curse those things up down low. You see what they look like anyway. The things, the dry erase markers dry before they and are hard. They dry easy and they erase hard. The chalkboards, those things get all kinds of, residual goop, and then they're a pain to wash off and stuff. So you see the side effects of that in the
Oh yeah,
I'll
in the
take,
lab.
yeah,
The lab
I'll take
one's
chalk. Yeah,
terrible.
I'll take chalk any day of the week. At least it's a lot more erasable than what I have.
Well, so there's good chalk out there. You
There
just
is, yeah,
haven't.
but Prang and crapola are not one of them. Are not too great. They're kind of like, it's like Yugos in cars, Okay. Well, I guess cheap, right? Yeah, that's the main thing. Okay, but anyway, RNA interference. Okay, what is that? Well, crudely, the quick and dirty of RNA interference is okay, the quick and dirty is that there are certain small RNAs, they're often called small interfering or siRNAs. And what they will do is they will bind to complementary sections of messenger RNAs. In the process, yes?
What is the word after or?
Or cause.
Okay.
When they do that, they either can block translation, or in many cases, they can actually trigger off destruction of that messenger RNA that they target. Now, that's really going to block translation. The mRNA is gone. It seems to be an important developmental mechanism. It's now estimated, at least in vertebrates, perhaps 50% of our genes, the expression of 50% of our genes, and possibly more, this is probably perhaps a lowball figure until they get more data, are going to be regulated in part by this RNA interference mechanism. RNA interference defects in it can lead to gross developmental defects. There's also some implications in human diseases. And they have artificially produced small interfering RNAs, have powerful capabilities in research and are widely used. And there is interest, in fact, I think one is in clinical trials now, and potential clinical aspects. Now, as a matter of fact, in my own personal case, one of several things I'm missing from chromosome 17 that I wish I hadn't been missing includes a gene that codes for only small RNAs that seems to affect the translation of certain proteins involved in regulating cell division, which is not the thing you really like to have. But, yes?
What's the improved genes in parentheses?
Oh, about 50% of our genes are regulated this way. So clearly this is an important mechanism. It was only discovered almost by accident about a decade ago. But since then, people have studied it extensively because of its potential implications. So let's take a little look at this whole process. We're going to start out with inside the genome. There happen to be, and in plants, perhaps 1,000 of these, in vertebrates, maybe 500, maybe more. There happen to be scattered throughout the genome, sometimes in introns, sometimes in regions between genes. They can be scattered all over. These small little short genes. And these little tiny genes are transcribed. These genes are only several tens of nucleotides long. And they get transcribed. I think most of them are polymerase II transcription, if I recall. Okay, but these genes get transcribed and you get these small RNAs, sometimes called microRNAs because they're quite small. Okay, now interesting thing about these small RNAs, they have what's called a palindromic sequence. Now a palindrome is a sequence that's kind of like, you have a sequence in one thing and you have an opposite one somewhere else. So let's just make a real cheap and simple one. Oh, that's a cheater's palindrome. But you see something like this. Obviously, sequence here, opposite or complementary sequence at the other end of that RNA. And what will happen here, of course, is these guys can hydrogen bond with each other. So these microRNA genes have these. So now we transcribe these microRNAs, and what you have is a single stem and loop-like structure with the palindromic part, hydrogen bonding, and then this loop coming out. Now once again, these RNAs are fairly small. They're only several tens of nucleotides long. Now we're still a nucleus, and in the nucleus, what happens is an enzyme complex. I'll just put nucleus over here. An enzyme complex, appropriately called dicer, starts to work on these guys. And dicer is appropriately named because it's a specific nuclease. It slices, it dices. And what it slices and dices cuts this loop and part of the stem section off of that little RNA. What's that?
Oh, the picture of...
Oh, yes, yes. Now, you remember, if you ever saw the film The Shining, that was that scene with Jack Nicholson coming in with the axe. That's where it came from. All right. Okay. For the YouTube guys. Okay. Thanks. Well, thanks to Jack Nicholson, right? All right. Now, that's a good film, by the way. All right. That's where it came from. Okay. All right, well anyway, that's exactly what dicer does. It cuts, and now we have this little short sequence here that is often called a small interfering, or siRNA. It's only 21 to 23 nucleotides long. It's a very narrow range of size of these guys. And you notice it is double-stranded because this was part of the old palindrome sequence.
No, that's S-I?
Yeah, small interfering. Okay, small interfering RNA. And they tend to be, like I say, the size of these guys is precisely controlled about 21 to 23 pairs of nucleotides long. So here it is. Okay, now we've done this. We've processed these microRNAs. We've got our small interfering RNAs. Now what's going to happen is we're going to take these guys into the cytoplasm. So away they go into the cytoplasm. That's where these guys are going to work. Once into the cytoplasm, they bind to a large enzyme complex. In fact, I'm going to let me move the cytoplasm over to here. So, is it okay if we get rid of this stuff?
I suppose you can move the cytoplasm.
Okay. Too much nucleus here. Okay, so we're in the cytoplasm now. All right, we're completely out of the nucleus, and now we are in the cytoplasm. Okay, our little small interfering RNA binds to a large enzyme complex that's known, abbreviated as the RISC, or RISC complex. And what it means is RNA-induced silencing complex. See why we have acronyms for these things in biology? Okay, and what that means is, overall, is that RNA triggers off the silencing or ramping down of the expression of certain genes. Okay, so here's our little siRNA here. Now this RISC complex contains numerous proteins, including one protein that's in a family called the argonaute proteins. That happens to be an RNase, in other words, RNA digesting enzyme, and several other proteins. Now the first thing that's going to happen is this RISC complex somehow removes one of those two strands of RNAs and holds on to the other. So we remove one strand. So here's the remaining strand, and of course what that means is the bases on the remaining strand are exposed. They're out there exposed. Stripped and gotten rid of the second strand here. So this sequence can presumably hydrogen bond to a complementary sequence if one happens to come along. And that's where this process happens. Now, let's suppose a messenger RNA comes in. And part of that messenger RNA happens to have a complementary sequence to that particular siRNA.
So it splits the siRNA after it binds to the complex?
Yeah, it binds, in fact, it's one of the proteins, one or more of the proteins in the complex that removes that second strand.
Okay.
Now we have a complex with that single strand of the siRNA, and those bases can now hydrogen bond to any complementary sequence it finds. So here's the messenger RNA. It's got a complementary sequence somewhere, and it's going to stick to the small interfering RNA. Now that complementary sequence could be anywhere on that messenger RNA. It could be part of the coding sequence, or it could be part of the untranslated region. Now obviously you don't want to do that with the poly A tail, because then this process would occur with anything that has a poly A tail, which would be an extremely bad thing. Okay, but complementary sequences can be on the coding sequence or the 3' untranslated region. So now this messenger RNA sticks, and that's going to take us to the next step. Now let's suppose we have an incomplete match. In other words, on these, say, 22 bases here, 18 of them hydrogen bond, and the others are not the proper base. So we have a partial match between the RNA and the messenger RNA. What happens there is we still have this double-stranded section here. Now let's suppose a ribosome grabs onto it and tries to read this thing. Ribosomes cannot read double-stranded RNAs, and especially if that happens to be complex to this big, huge enzyme. So what happens, the ribosome reads down this thing and then runs into this and kiss off translation. It stops at that point. So what's going to happen? Blocks translation, and then you get to that double-stranded region, translation stops. Now the second thing is slowly that small interfering RNA and that messenger RNA will slowly get released and shipped to these regions in the cytoplasm that are called P bodies. And this is a slow process. I don't know what that P stands for, by the way. P bodies are regions in the cytoplasm that can best be described as graveyards for unwanted, unloved messenger RNA molecules. Because inside the P bodies you find they stain intensely for RNA, showing there's a lot of RNAs in there. And there's numerous these speckles of these P bodies in the cytoplasm. And they also are full of nucleases. Apparently they're attached to some type of scaffold that I don't think we know the details about that, but somehow these P bodies are sites where you have a lot of nucleases there, RNA digesting enzymes, and RNAs that are no longer loved or wanted get shipped out to XL and a slow death in the P body extermination camps. Okay. All right. Modeled after certain people about 70 years ago who were very good at that type of stuff. Okay, so P bodies are where we get rid. They're RNA degradation centers in the cytoplasm. Okay, now let's suppose we have a complete match instead. In other words, all these bases are complementary to each other. That's a little bit of a different story. What happens is somehow that activates the nuclease activity, these Argonaute proteins. And both RNAs are destroyed. So once we activate this nuclease, we take out that messenger RNA entirely. We also remove the siRNA. And now the RISC complex is back waiting for another one to bind. Yes?
That's Argonaute?
That's what it's called, yeah. And it's spelled Argonaute, I guess French, you know. Same people that make this cheap job. What do you expect from French people? They cheap job, they misspell words. You know.
Now we're going to make...
Yeah. Okay. All right. So, in other words, what we have here, incomplete match means you get sent to the concentration camp and a slow, miserable death. Complete match, it's quick. It's quickly over with. You're wiped out very rapidly. The end effect, though, of this process is the translation is blocked or strongly inhibited. Now, why do we do this kind of thing? This gives us another avenue for regulation. So why do we do this kind of thing? Okay, well, let's suppose we have produced, we're transcribing some gene. That gene codes for a stable messenger RNA. So now we got a whole ton of stable messenger RNAs in the cytoplasm. And now at a particular point in development or a particular point in particular environmental conditions the cell is exposed to, whatever the case is, now all of a sudden we need translation of that gene blocked quite a bit. Right? Okay, if you have a ton of messenger RNAs for that code for that protein around, what do? They're floating around for a long time. You've got to get rid of them one way or another. One way of doing this with this RNA interference mechanism. You start taking these guys out one by one, and eventually you dropped it down to whatever level you want. So that's one kind of thing. Get rid of messenger RNAs that we don't particularly want. We want to ramp down translation, expression of that particular gene, but we've got stable RNAs in the cytoplasm. Let's get rid of these guys. Another thing where some of this mechanism may have evolved from is what are called transposons. Okay, now transposons are sections of genes. Somebody's heard of those guys before. They're particular DNA sequences and they have an ability to get cut out of a stretch of DNA and move somewhere else. Jumping genes, they've often been called. Now, Barbara McClintock, she died many years ago, spent her whole career at Cold Spring Harbor studying odd aspects of corn. She gave cornfields out in the back of the lab facility. And she, using classical genetics, found that certain genes could shift. There were certain genes that could shift their location in the genome. And called them transpose. A lot of people thought she was a crazy old lady, but her evidence eventually became so so solid that you had to accept that there were these, quote, jumping genes around. She got a Nobel for her work later on. But anyway, what a lot of them will often do is they will copy themselves first and then move the copy over. So now you have two of them instead of one. So on. So sometimes you have these sequences. Now they may have been derived from viral sequences. We don't know how these guys got in the genome. It's possible that they're derived from viral sequences. But the problem with these transposons, especially ones that do the copy and paste function, just like you do on a word processor, you keep the original copy and you make another copy, you put it somewhere else in the sentence. Okay, now, what's going to happen? It means you're going to get more and more and more of these things. And that can eventually start causing serious problems in the genome. Especially, most of these guys end up transposing themselves randomly. So what happens if you end up sticking one of these guys right in the middle of a gene that's absolutely critical for life? Well, the cell's going to die. Or you separate a gene from its regulatory sequence. Who knows what that's going to do? Okay. So, and after a while, if you keep on doing this stuff, you can get so much of a genetic garbage load that it really starts slowing things down, becomes detrimental to cells. As a matter of fact, there are some sequences, and one of the common ones are known as the line elements that appear to be these types of transposons. They're short sequences, I think a few hundred nucleotide bases long, but there are millions of them in our genome. They're all over the place. Okay, now how do you help protect yourself from these things? Well, many of these, especially the copy and paste type, also will code for what we can sometimes call an integrase. Can't spell it. An enzyme that actually can do the cut and paste function. Okay, one way of controlling these guys would be if we could block the expression of that. Then the things would be sitting there unable to duplicate and unable to transpose themselves. So if we can block the expression of things that are needed for this kind of transposition. Another thing is most transposons have palindromes in them. So there seemed to have been an evolutionary push to come up with mechanisms to deal with too many transposons. We can take a little bit, we can take some of that stuff. But what we can't do is have too much of it. Then we start getting overload, and it starts causing problems. So we got to keep these guys somewhat in check. So, yeah, palindromes? How about an enzyme that recognizes those palindromic sequences and cuts? Dicer. Okay. How about a mechanism to stop the expression of selected genes? Could be that small interfering RNA. So it seems that this whole thing may have developed first as elements of it as protection against too much problems with transposons. And then later on started playing regulatory roles in cells. All right.
Why did you write line?
Oh, that's the name of those genetic sequences that are duplicated millions of times in the genome. They're called line elements, and they're believed to be essentially derived from transposon. And if you do genetics, if you take molecular biology, you'll probably mention these. And they're literally duplicated millions of times in our genome, all over the place. Genetic garbage. Okay, so that was just one name of this kind of thing. It's derived from transposon. It just fills up our genome with trash. Like my mailbox when I come back home, it's all kinds of garbage in it. Okay, all right, so that's some hypothesis about the origin of this, but now it's being used for regulatory purposes as well. RNA interference was first discovered in plants. And once they had an idea partially how it worked, they could do things like, what would happen if we deleted the gene for Argonaut, or we deleted the gene for Dicer? In other words, we stopped the whole RNA interference mechanism. Well, they did that in plant embryos, and what they found is gross abnormalities as the plant grew. They found certain tissues or certain plant structures either vastly undergrew or vastly overgrew. Like you might have excessively humongous leaves that are so big and so heavy that the plant can't support them. Or maybe a little tiny stem, we need a much longer stem. Things like that. You can do all kinds of things. And what a lot of these things appear to happen is regulating effectively the growth of certain tissues and certain organs. Now when they started doing the same thing in animals like mice embryos, they found similar things. Over or undergrowth of certain tissues and organs. And sometimes the size of the organism also was either too small for normal or too large for normal. So a lot of times what we see in development is RNA interference in many cases seems to control and coordinate the relative growth rates of certain tissues, organs, or what have you.
So it basically works the same in plants and in animals.
Yeah, it's just different kinds of tissues. But it was first discovered in plants, and then they said, hey, do animals have it? The answer is yes. Okay, so we have these kinds of things.
Wait, RNA interference helps to what?
Oh,
Help to-
in a sense, kind of coordinate growth of tissues and organs so you don't have certain things overgrow or undergrow. Like for instance, you would be perhaps in very bad shape if your liver grew to the relative size of, say, a shark liver. Imagine having a 3-foot-long, 30-pound liver in you. That might not be too good. And, or undergrowth of certain parts. Oh, we got this little tiny heart here to develop things today, but it's not enough to pump enough blood in me, so I'm going to die. Okay, well, it's cute, but it's not going to sustain me. Okay, so you don't want relative over or undergrowth of tissues or organs or what have you. Or, you know, oh, my brain's this size, I'm going to run for Congress. That explains a lot, right? All right. Okay, well, so a lot of that does seem to be regulating various kinds of, especially perhaps many kinds of growth in it. We're thinking, even though our little lab manual is about 10%, the figure now of genes are regulated by RNA interference is more like 50% now. That's a reasonable estimate. So it's obviously very important. Now, one thing we can say here is one siRNA a single siRNA, depending on what the complementary sequence is, could regulate just a single messenger RNA, where you have that sequence and it's not found anywhere else. Or it could regulate numerous RNAs, where you have some kind of sequence that is in common. Many of the ones that probably target some of the untranslated regions are probably going to be able to deal with many different genes, because there's going to be more similarity perhaps in the untranslated regions than there are in the actual coding regions. So depending, a single small interfering RNA may regulate the expression of a score of different genes, or it may regulate the expression of a single gene. In fact, that's one reason. Now, we're pretty sure there are at least 400 or 500 microRNA genes in our genome. There could be many more. It takes a while to look and say, hey, what is this sequence? Just by looking at, genetic sequences, trying to identify what sequence are actually code for this gene or that gene is actually a difficult process. So we don't know for sure, but we identify about 4 or 5, and plants have at least, angiosperms have at least 1,000. It's probably more than that. Okay, now, on the other hand, you know, if you're talking about, say, 1,000 genes for these guys, and you're regulating perhaps 10,000, the expression of 10,000 genes, well, that's pretty insignificant. That means that most of these guys are going to be regulating several different genes at the same time. Okay, and that actually can lead to some potential problems later on. We'll see that in a little bit. Okay, now, clearly developmentally important. But, oops, I think, all right, just realize what time it is. Okay, I think we'll take a look at applications and other aspects